Good morning, all. Dr. Pete here. I am a PhD biochemist and also a nutrition network health practitioner. And I apologize to my viewers for being rather absent lately. I am busy right now in another location besides where I live, doing a 30-day live-in with a client who is actively reversing diabetes and is struggling with sugar addiction. Is metabolic disease a maladaptation of normal metabolism? Now, I've been working in this area for 5 years now. I reversed my own prediabetes, and I also put my gout in remission. And the skin that I have in this game is in an effort to try and understand why metabolic diseases happen so that I can be better at helping people recover, put them in reversal, and put them in remission by knowing and understanding the processes that lead to the disease in the first place. I do not have my own laboratory. I have no way of carrying out these clinical studies myself. I am not able to do the bench work, the in vitro, the in vivo work myself. Many would call me now a theoretical biochemist. I have found, and I've been interested since the beginning of my own journey 5 years ago, when we see influencers in this area, medical doctors in this area, the coaches that are in this area talking about reversing chronic diseases, and they talk about insulin resistance. I have asked the question since the beginning, since I got into this. What is the root cause of the insulin resistance in the first place? What is driving these metabolic diseases? And we're going to be talking about what I believe is the fundamental mechanism, at least one of the fundamental mechanisms today that drives these diseases. And before we get started into this, I just want to remind everybody that I will be in collaboration with the Nutrition Network offering a masterclass in the area of gout on the 9th of March. And I put the link for the registration of for that masterclass in the notes in the description. And I would like to point out to the gout sufferers that normally listen to me, this discussion this morning is also for you because and I'm going to get into this. We might as well get into it now. One of the driving factors of gout is, in fact, has to do with the mechanisms that we're going to be talking about this morning. And these are the mechanisms that sit underneath the concept of insulin resistance. It's not the only one. And I'm not claiming that this is the whole enchilada of insulin resistance, but I am making or taking a stake in communicating this is one of the main drivers. All right, so let's get started. And I want to do that with a quote from a recent paper that I've been immersed in even through today that was written by McCarthy in 1965. And this has to do with why people come down with gout. And he pointed out, based on this philosopher, that if we don't look at the past, right, if we're not willing to go outside our hallway and to study the questions that we have by reaching out into different disciplines, we cannot come up with the answer. We need to be careful about existing. And this is the language that we see a lot in I think, as a criticism towards the space that I'm into, that we should stay in our lane. Right. Well, here's the problem with staying in our lane when we're trying to answer a significant question about human existence and human ability to live. Right. If we stay in our lane, what this does is it prevents us from answering crucial questions. And one of the questions that we have is what causes chronic disease, right? What causes type 2 diabetes? What leads to and causes obesity? What ends up causing things like gout? All right. And if we want to have answers, if we want to find headway or push through the headway to come up with answers about these crucial issues, we've got to be willing to look at all the different aspects of our biology in order to answer, or at least to come up with some of the solutions to these problems. So one of the issues, and this is a gout slide, but it's the same for all chronic diseases, right? There's a stigma attached to this. And one of the things that we need to really do, not only as coaches, but as people that are suffering from these diseases, is find a way to get past the stigma, right? It's difficult to talk about these things. It's difficult to talk about them even with family members because they have a tendency to look down their nose at us and feel like we've done something deliberate to get to where we're at. And for those of us that have really delved into these issues, we know that that's not true. The standard American diet is, in fact, along with its nuances, is in fact the driver behind these diseases. I feel very confident in saying that. And then basically one of the components that's in that way of eating is the overconsumption of sugar, and mainly one of the components of that being fructose, right? That's probably the biggest issue here. And you'll see that as we get into this, right? Because it's not just the fructose that you're eating off your plate as a metaphor. It's also the fructose that's being produced in the cells where we have fructose uric acid metabolism. And even as I say fructose uric acid metabolism, I say that carefully because I am not going to live in the hallway of just fructose uric acid metabolism. One of the things we need to do today is to learn how that metabolic pathway is integrated with other pathways, and the maladaptation, the maladaptation of that pathway to the way that we now eat in 2023 is what's driving these diseases. This diagram is making the point that not any of these diseases, whether we're talking about gout, kidney disease, diabetes, obesity, Alzheimer's, as a matter of fact, cardiovascular disease, and sugar addiction, by the way, which is a biochemical brain disease, right? These all have a common denominator in maladaptive metabolic processes that are connected in a powerful way to gene expression in the brain and a specific protein there that's called delta phosphB. Now, in today's discussion, we are not going to focus on delta phosphB and what it does. We are going to talk about the biochemical pathways that are involved in this maladaptation that is central to all of these diseases. This is just to point out what I'm talking about in the past slide, the fact that these things are integrated, and it's very rare that somebody suffering from one disease is not also suffering from other comorbidities. Like in the case of gout, over 74% of those individuals are hypertensive. And for 5 years, I've been working with type 2 diabetics, and I know damn well that those guys are also suffering from hypertension. As an example, kidney disease is very common. Over half the individuals that suffer from gout also are also significantly obese. A full quarter are type 2 diabetes. This is a problem that exists for all of us, especially those of us that are older, and we've been living and eating on this planet long enough, right? Decades and decades of the standard American diet, which is high in sugar, so that we have gotten to this point, right? I know that in my own collective group of friends who are my age, everybody seems to be on medications. Quite a few of them are talking about the fact that they're not planning to be around in their 70s very long, right? They just want to check out, when in fact there is something that we can do about our health status. We can make a decision about what we're putting in our mouth, and that's going to matter here. Again, understanding the biochemical drivers of these collective diseases is super important to us, because we can reverse or put in remission these conditions by changing how we eat. This is a very important thing. Not all of us suffer from the same comorbidities, right? There is this factor of genetics involved in this, and how we respond to the environment, and the food environment, the exercise environment, lifestyle environment, right? All of those things influence what ultimately is going to happen to us from a metabolic point of view, right? And we're all individuals, so we end up on an individual basis arriving at a different set of these comorbidities in particular, although we can statistically be broken into groups. And my cross categories were high blood pressure, number one, prediabetes, number two, and gout. Those were my three. Now, over the last 5 years, I've reversed the prediabetes. I have put my gout in remission. And additionally, my blood pressure now is significantly lower. I'm going to say normal. So on this first slide, what you're looking at is the combination of fructose uric acid metabolism in red and the glycolysis pathway in gray on the left side of the slide. This business is not all about the glucose, right? And we hear about this glucose-centric point of view about diabetes all the time. On the news, when we talk to influencers, we hear, okay, it's the insulin resistance, down the road we go, and how the glucose is being processed. Okay, that viewpoint is partly right, but we cannot look at glucose metabolism in glycolysis, and we cannot look at fructose uric acid metabolism. The key thing to understand going into this is that these pathways are integrated and they operate in concert with one another. And there is a relationship between how much stuff is coming in and how much macronutrient can be handled at any one time by a given platform and or pathway. So let's start out first and just summarize what's going on with glycolysis. Again, that is in the gray pathway represented on the left side. So the main purpose of glycolysis is to accept the input of glucose that's coming in under normal concentrations of glucose. And some of you out there that are listening to this, you may want to bust me out right away on that, because you're going to ask, well, what's a normal amount of glucose coming in? And honestly, finding the answer to that, good luck if you can. A lot of the ways that these experiments have been done have been very laboratory manipulated. In other words, they've chosen a certain concentration of glucose to work with. Then they've done their studies and they've based their data, the results that they're seeing based on that. And one of the key areas that we don't understand here going into this is what the threshold amount of glucose coming into the liver, and that's the model system that we're going to be mainly talking about here. So we eat a meal, the macronutrients are going to get processed through the intestine, they're going to arrive into the portal vein, which is the blood that is feeding both interacting with the pancreas, and then the main clearinghouse is the liver before the end products of all that business go out into the peripheral circulation, which would be bringing things like glucose and other nutrients to the skeletal muscle tissue, adipose tissue, brain, and so on. So remember, to reiterate, the purpose of glucose, or excuse me, glycolysis, is to provide the cell with energy, ATP production. Glycolysis is also connected to the mitochondria, and as long as we have a normal flow of glucose coming in, then we are going to have and this pathway has checks and balances, and it has feedback mechanisms and so on, because the main purpose of this pathway is to produce energy, to store some of that energy, and to provide inputs for the mitochondria, where we get the majority of ATP, the energy currency of the cell. Now, when somebody sits down and they have a standard American meal, like if we focus on the dinner hour, and maybe you've got somebody who's gone into a bar and they've ordered a couple of beers, right? They've ordered a large sausage pizza. They've ordered the breadsticks. They've ordered a salad with commercial dressing on it. they have a meal at that point, which is hyperglycemic, high in glucose, no longer in the normal range, right? So what's going to happen at the level of the liver is that glucose is going to build up. It's going to start to pool in front of the glycolytic pathway. The glycolytic pathway is going to come to its capacity relatively fast. We have incoming sugar, right? Added sugars from a meal like that will have fructose. So we have exogenous fructose coming into the meal. And then, as I said, they also have a couple of beers. So you're going to have not only high carbohydrate there contributing to hyperglycemia, right? The glucose load coming in. Some beers have been brewed in high fructose corn syrup. So there may also be a fructose load coming in. And then we also have the alcoholic component. And that is fundamentally important because in this meal now, which has what I call the deadly triad, hyperglycemia, fructose, and also alcohol, we open fructose uric acid metabolism via the polyol pathway, whose first enzyme is called aldose reductase, which is activated by the increased glycemia and the alcohol. And what happens there is that much of that glucose that's pooled at the top of glycolysis is now funneled into fructose metabolism. So we start actually producing fructose directly within the cell. At the same time, we have fructose, I call it exogenous fructose, coming in from the meal, which is also delivered into the liver, which joins the fructose that's already in there coming in from the hyperglycemia. And now we have fructose metabolism. And you can think about the fructose uric acid metabolism the following way. If we step back and we think about the glycolysis first, with normal glycemia coming in, this is like having a dam that is pushing water back up against the dam. So we have pooled water behind the dam. And glycolysis can be thought of as the stream from the bottom of the dam, and it is proceeding in a nice even flow. And you can think of that as generating the cellular energy that we need. Some of it is going to be stored power. You know, the water that is kept behind the dam is providing stored energy and so on. We might, if there's enough glucose, but not beyond this threshold into the hyperglycemia realm yet, then we may also be storing some of that energy, a little bit of that energy as fat. Now, as we go to the hyperglycemic load and we bring in the external fructose and we bring in the alcohol and we activate polyol pathway, this is like opening up the floodgates of that dam, right? When the water level gets to be so high, they've got to let water out, because if they don't, it's going to risk the integrity of the dam. And that's when those big old jets are just flying, right? And the water is just coming out, because the fructose uric acid pathway, once it gets opened, is essentially, in one way of thinking, it's all for none, right? It's just running. It doesn't have the feedback loops. It doesn't have the checkpoints. This is all for nothing. And the consequence is, in summary, is that we see a very steep ATP decline along with a phosphate decline, at the same time having an acute significant rise in uric acid. And uric acid on an intracellular basis being produced in this manner is a signaling molecule which causes an elevation in systemic inflammation, disruption of the nitric oxide pathway. Think downstream in the endothelial layer of the cardiovascular system, and disrupting cardiovascular function. And then lastly, we have the liver is basically reprogrammed into fat storage, into de novo lipogenesis. So that we have the mitochondria is downregulated. So this is another diagram of exactly the same pathway, but from a slightly different point of view. So at the top on the right, you can see glucose being converted into fructose via the polyol pathway with an indication of the things that activate that pathway. So things like alcohol, for example, high salt meals, think processed food, right? Um, Osmolarity differences. This has to do both with the salt and the alcohol. When we bring those into the blood supply, this changes the relationship between the water and the things that are dissolved in there. And the polyol pathway is sensitive to this. And it's sensitive to it because, and this is sort of stepping back, evolutionarily speaking, the processes that I'm talking about here were super important to the survival of humans back in the day when we were hunter-gatherers. And our food supply was not guaranteed from day to day, right? So what we're talking about here is a very interesting phenomenon. You can read about it by Dr. Richard Johnson, for example, or you can see his presentations on YouTube. And he characterizes the processes that we're talking about here either as the survival switch or as the fat switch, because this is a major driver of obesity. But going back to evolution, this was important because when humans came on a food source, we were able to store very effectively the inputting calories that we were eating into fat and into water. All right. And you can see that here in the conversion of glucose to fructose by the polyol pathway, because the activators here, the ethanol, the salt, the ethanol partially. There are other ethanol effects which we're not going to get into in this presentation, but in part, the ethanol, the alcohol component of these drinks is affecting this osmolarity. So is the high salt of processed food. And then interestingly, hypoxic environments, and this is where sleep apnea, the effect where people during the sleep cycle actually can stop breathing for short time periods or increments, causing hypoxia to occur intermittently through the night. And hypoxia activates this pathway. And if I get a chance, I'll come back and talk about that a little bit more. Now, in this slide, we can see, in particular in the middle, the effect when this pathway fully opens up. So the floodgates are flying. We have the hyperglycemia, we have the alcohol, we have fructose coming in from the outside, we have fructose being produced endogenously within the liver itself. We see that in the very first step of conversion of fructose to fructose 1 phosphate is where we get this substantial energy decline. The ATP is used to phosphorylate the fructose. It is concentration dependent on the fructose. Happens in a very short period of time. So we decimate the ATP concentration in the liver. We decimate the concentration of phosphorus in the liver, and we have this acute sudden rise in uric acid. We also have the activation of AMP deaminase. And this is going to be important to us on the next slide when we talk about the influence of certain types of protein that has high quantities of the amino acid glutamate and also provides inosine monophosphate and adenosine monophosphate that the liver is going to see, because this is also another activator of fructose uric acid metabolism. Now, one of the main points of this slide that I want to make sure to emphasize before we move to the next one is that we don't just have biochemical effects happening in the liver, the systemic inflammation, the de novo lipogenesis, the disruption of nitric oxide. We also have an influence on hunger and thirst. So this pathway, when it's opened, increases our hunger, increases our thirst. It promotes compulsive eating, and it causes a shift in the liver to fat synthesis and storage, or oil droplets actually forming in the liver, which can lead to non-alcoholic fatty liver disease. One of the main purposes of this slide, and hopefully you're keeping up with me right now. So we have this person who's eating the standard American meal, hyperglycemic. Alcohol could be there or not be there. It depends on what meal of the day that it is. We have added sugars coming in with the fructose component. We also could be eating something which is high in a glutamate source. This has an effect on opening the pathway. I want to emphasize really are two things. The effect that when this overall integrated pathway, right? We're not just fructose uric acid metabolism. We have a relationship here between glycolysis, which is overloaded, fructose uric acid metabolism. And in this slide, I want to talk about two things. The first is we have significant activation, fatty acid synthesis directly in the liver. And we see that by the two transcription factors that are in yellow, the carbohydrate regulatory element binding protein, and you see over there sort of central right, the sterol regulatory element binding protein 1C. Both of these transcription factors are activated by fructose uric acid metabolism when this pathway is wide open. Additionally, we have an insulin response here. Both of these transcription factors are also influenced by the level of insulin. So we're getting a double whammy here. And these two transcription factors are opening de novo lipogenesis, all right? And you can see that the lipogenesis, the production of the fat, if you look down on the slide, lower right-hand side, you can see the citrate, 3 red arrows going to the triglycerides and then the VLDLs. We want to talk about briefly about the mitochondria here because the uric acid that's being produced, right, we've already talked about that has a significant effect on the downregulation of the burning capability in the mitochondria. So the uric acid causes the translocation of NADP oxidase into the mitochondria, which has the effect on downregulating an enzyme called aconitase, which is the enzyme that catalyzes the reaction between citrate in the mitochondria and isocitrate. So when aconitase gets blocked, now the citrate that is accumulating in there is shuttled back out into the cytoplasm, and that's now fatty acids cannot be transferred back into the mitochondria. So basically what we've done is we've reprogrammed the mitochondria and therefore the cell into the storage mode, the production of fat, and then the shipping of fat, well, the production and storage in the liver, and then the shippage of fat out of the liver. And then I cannot forget, over on the right in red, we see that fructose uric acid has two significant effects on the effect of insulin. Number one, the fructose uric acid metabolism affects the insulin receptor substrate proteins 1 and 2, which interrupts insulin signaling at the level of the membrane. Thus, the insulin coming in is not delivering the insulin signaling at a certain point. Thus, we have insulin resistance getting generated downstream of this. And then the other thing is we also, there is data to show that the insulin receptor itself is actually decreased in the membrane of the liver. And so this also is going to contribute to the effect of hyperinsulinemia, high insulin, and also insulin resistance. And it raises a question that I so far cannot find an answer to in the literature. And that has to do with insulin extraction at the level of the liver. And the liver is responsible for reducing the amount of insulin that reaches the liver from the portal blood supply due to the secretions of the pancreas. And anywhere from 50 to 80% of the insulin would normally be removed and not reaching the peripheral circulation. So in this case, we see reduction of the receptor at the level of the membrane. This is going to have an effect on the amount of insulin that is passing by the liver and reaching the peripheral circulation. And it raises the question about what is the regulatory effects on insulin extraction, which now I'm going to leave behind because we need to move on to the next thing. So this brings me to the third and really the final slide of this presentation, besides what we can do about this. So you are looking at something that is now becoming more and more common when you look in the literature at a particular tissue that is undergoing the kind of biochemical transformation with fructose uric acid metabolism that we are talking about here, and that is the Warburg effect. So everything that we've talked about is still true in this slide. One of the things I've added to this is that when fructose uric acid metabolism is activated, it is operating at a rate which is 10 times the rate of glycolysis. So again, thinking about once we've activated this pathway, the floodgates are open, right? And the glucose is just being pushed off into this pathway. Fructose is coming into it. And we have this very high sudden acute rise in uric acid, which is a signaling molecule that it's eliciting effects, not the least of which is causing the liver to move into the fat production mode, the downregulation of the mitochondria. Now, the Warburg effect, this is an effect here. If you think about what's going on, the dihydroxyacetone phosphate and the glyceraldehyde that I show here in turquoise, this is an important intermediate in fructose uric acid metabolism. And because this pathway is operating at 10 times the rate of glycolysis, you're going to have a pooling effect of the DHAP and the glyceraldehyde, which are now going to be converted into substrates in the glycolysis pathway, but they're cycling in lower. One of the consequences of this will be for the production of an excess of lactate. And guess what? Lactate, like uric acid, is also a signaling molecule, and it has mitochondrial effects. Predominantly when it rises into higher levels, it causes the downregulation of a receptor in the mitochondria called CPT2. So this has a double whammy effect on the ability of the mitochondria now to oxidize fatty acids, because we have knocked down another mode of entry where the acyl-CoAs would be funneling into the mitochondria through CPT2, but now because of the elevated lactate cannot. There is another effect here as well, and that is that as the lactate pools and we have shut down the mitochondria, cellular acidosis is increased, the amount of hydrogen ion, as lactate is pushed out into the circulatory system because of its elevated status. As it's pushed out through a monocarboxylate transporter, it is symported. In other words, it's kicked out into the circulatory system in a 1-to-1 ratio with hydrogen ion. Hydrogen ion is also pushed out into the blood supply by other mechanisms. This is really important to gout sufferers, because there is evidence that in the joint of a gout sufferer that the chondrocyte undergoes this type of metabolism, and the acidification of the synovial fluid around the chondrocyte causes the release of calcium, which is needed in order to nucleate the crystals that form, the monosodium urate crystals that form, that can, in combination with the innate immune system, cause a gout flare. So to finish this, what we've learned is that when we hyperactivate these pathways, that this can lead to a state which, when humans were evolving, was a good thing, because we needed, when we got on a food source, our biochemistry, our needs as an organism required that we have very efficient ways to store fat and water, because when we finished with that food source, it was unknown how long it would take to get to another food source. So we needed an efficient way of being able to store these macronutrients so that we could utilize them by burning between leaving that food source and arriving at another. Our biochemistry was not prepared for year 2023, where we have chronic activation, hyperactivation of these pathways, the overload of glycolysis, the funneling effect into fructose uric acid metabolism, the downregulation, the chronic downregulation of the mitochondria, the de novo lipogenesis, the production of oil droplets in the liver, leading to non-alcoholic fatty liver disease, dysregulation of our glucose homeostasis, as an example, the onset of gout in some individuals, cardiovascular disease in others. So how do we combat this? There's really good literature going back into the mid-1800s, if you're willing to look. They knew back then that we could reduce the onset of these chronic diseases, or we could reduce the severity of them by eating a mixture of animal and vegetable foods. If we're suffering from significant comorbidities, so for example, we have type 2 diabetes, we're significantly obese, maybe on top of that cardiovascular disease, then the literature is really solid on this. By reducing the refined carbohydrates, stopping the sugar, and then cutting into the carb load so that we're eating no more, at least initially for the first few years, that we're only eating 50 grams of total carbs or less per day, that we can reverse these conditions. We have protein modulation, making sure that we're eating a wide variety of different meats and fish and so on, that we're not stuck eating one type of protein every single day. We eat green listed vegetables. We're still eating carbs. Granted, some people listening to this may be thinking, this guy is what, 0 carbs? I didn't say that. Let me be clear. I said we were reducing the carbs. And I, for 5 years, have been eating pretty close to 50 grams of total carbs per day, not 0 carbs. Some people may choose to go on a carnivore diet. And for some people, that way of living is best for them because some of these vegetable products that they were eating were causing them problems. So a person has to basically take a very realistic look at where they are lifestyle-wise and then make common sense decisions about potential eliminations. In terms of fats, yes, when we reduce the carbohydrate calories, we increase fats. And I'm a full believer in making sure that we include a wide variety of natural, unrefined fats in our lifestyle. I think if you are suffering from significant comorbidities like type 2 diabetes, it's a really good idea to measure your biomarkers, weight, blood pressure, blood glucose, ketones, and including uric acid. And remember, if you are on medications when you start this lifestyle change, you need to be talking to your doctor. I am not a medical doctor, and no doubt one of the big factors that happens, especially with diabetics that are in this lifestyle, is that they end up usually having to change the level of their medications. And that's something you should only be doing in a conversation with your doctor. Now, with that, I'd like to close. I hope everyone's having a good day. I apologize for being absent again, but as I said a little bit earlier, I'm in a 30-day live-in with a client, helping them reverse diabetes and dealing with sugar addiction. And as a result of that, I just have not had time to do any of these presentations until today.